Good afternoon and welcome. We are a visual species. The way we interpret the world is highly dependent on how the world is recreated by our nervous system. A remarkable amount of our brain is devoted to the processing of visual stimuli, from receipt of that information into the eye through the retina, converting light into nerve signals and reinterpreting those signals into a stereoscopic representation of the world around us. Our language, our society, our culture have been to a large part built around our visual capacity. The loss of vision also has profound influence on how we can function in society. Retinal diseases such as retinitis pigmentosa or age-related macular degeneration are leading forms of visual impairment and blindness in a Western society, with macular degeneration, for example, estimated to affect more than 30 to 50 million people worldwide. It is therefore my great pleasure to introduce, as part of our clinical translational seminar series, Dr. J Garrett, a physicist and research fellow at the University of Melbourne. David is part of the Bionic Vision Australia team that produced one of the most exciting scientific developments in recent times. This highly specialised Australian multidisciplinary team, incorporating a number of research groups, physicists, engineers and surgeons, were involved in bringing Australia's first bionic eye implant last year, a world first. The second stage of the human trials have now commenced. David's team at the University of Melbourne Materials Institute is responsible for developing a high acuity device based on diamond to act as the interface between bionics and the human nervous system. A former competitive ballroom dancer and high-end London caterer, his story, along with the story of the Australian bionic eye development, will undoubtedly be fascinating. Welcome, David.
Are we on? Where on earth did you dig that up? It certainly was not in my bio that I supplied, so there we go. Good research. Yes, I am David Garrett. I do work in the Department of Physics at the University of Melbourne. My fundamental training is in chemistry, in particular electrochemistry, which I completed my PhD in at the University of Canterbury. So I was brought over in 2010, just at the start of the BVA consortium experience, particularly for my expertise in electrochemistry and interfacing materials with, at that time, bacteria. And so it was a bit of a hop to start making electrodes to interface with nerves as opposed to bacteria. Right, so I'm going to talk a bit about as I understand there hasn't been a BVA talk to you before at the WEHI. Great to be here, by the way, my first trip over in 4 years, so good to meet you all. I'm going to talk about the wide view device, which was the first device and the one that's done the clinical trials. I'll do a short introduction to that so you're aware of what that device looks like and what it can do, and talk about the high acuity device, which is meant to be the next step on from the wide view device. Right, bionic retinas. So if you are to recreate the sensation of vision in a person through electronic means, there's a number of areas where you could potentially target. The most obvious one is the retina itself. But what that relies on, of course, is that the patient still has a retina and that some function in that retina remains. So in fact, if you're going for a retinal implant, there's only a couple of diseases that are really going to respond to this kind of treatment. And the two top contenders are retinitis pigmentosa, a genetic disease which affects the retina, but specifically it affects the colour sensing parts of the retina, so the rods and cones. So your rods and cones down this side of the retina tend to die out, but your signal transduction facilities, your ganglion cells and bipolar cells reaching up to your optic nerve and so forth back into the brain, still function relatively normally, especially for people that have developed these diseases. Oh, thank you, a clicker. Clicker. And it works. Pointer. There, thank you. Right, so here's our retina translated over here. So the signal transduction equipment is still working in patients with this disease, which means you have active neurons in order to stimulate. So for a person, for instance, who might have suffered bomb damage or something like that, their eye is obviously going to be ruined and this approach will not work. You can potentially stimulate in the cortex, which it turns out is spatially mapped to the retina. So even though you might imagine that going to the cortex would be an insurmountable problem, you still actually have basically an XY coordinate system even when you go to the cortex. So there are a number of groups that are following exactly that approach and building stimulation arrays that can go directly into the cortex. In terms of where you put your implant, you can take a number of approaches. You can put it on top of the retina, epiretinal. You can put it under the retina, which is usually called subretinal. Or you can go in this space here, which is usually called suprachoroidal or subscleral. So the two devices I'm going to talk about today, the first one, the wide view, goes suprachoroidal, and the one that I was involved in goes epiretinal. The scleral pocket here has the big advantage that it's a very secure location. It kind of sandwiches the device in. The sclera itself is a very tough piece of tissue, so it's just a good anchor point. So devices that are in there tend to be quite secure. It also means you can put a device in there and not actually penetrate all the way to the intraocular space, so that you've got less potential for leakage, less infection risk. Going into the epiretinal location, you are much closer to these ganglion cells, so your potential for high resolution is better, but you have the problem of getting your device to stay on this piece of tissue, which is always ubiquitously described as being like wet tissue paper, extremely, extremely delicate piece of tissue. So interfacing with that is a real challenge, and probably, as I go through the talk, the toughest challenge that we face in building our implant. Stimulation on the retina, as you might imagine, you have an array of electrodes. When you have a single electrode and you just put a small pulse of current into that electrode, you can affect the chemistry in the solution immediately adjacent to the neurons, and you can get a ganglion cell to fire. If you can get one ganglion cell to fire, there's the potential that that will cascade, and you'll have a neural signal that will head off down your optic nerve to your visual cortex, and you'll perceive that electrode as a spot of light. The spot of light is generally grey in colour, or white, or sometimes yellow, depends on the patient. That spot, as we found out through the clinical trials, is not in fact usually a spot. It can be a streak, a bow, a donut, a puff of smoke. All sorts of various things can occur. So a lot of the people, the very smart people that were developing maps of how you might use this information at the start of the program, were making arrays of little spots and imagining how you might present image with those arrays of spots, had a huge challenge when it came to trying to recreate that whole concept to these blind patients not using spots, but using a collection of bananas and puffs of smoke and rainbows and little lines. So yes, an interesting journey, and obviously still in its infancy, so a lot to be learned in this field. That is the suprachoroidal implant. It's called the wide view device because its electrode array is relatively big and wraps around the back of the eye, so it gives you a wide field of view, essentially. Your electrodes are spread across quite a wide space. Wrong one. Stimulating electrodes, 24 of them. 2 large counter electrodes. This is a helical platinum wire cable. A bend there. This little bit here just sits in the orbit of the eye, locked into the bone, and this cable goes onto the device. A reference electrode here, and this was a percutaneous plug. So when the grant for this was really written, the whole thing was always intended to be wireless, but very quickly we realised that we were just so far behind the rest of the world in this race that they needed to get a clinical trial underway as soon as possible. So rather than waiting for the electronics to catch up for the package that would control it all, the simple solution was to take this percutaneous plug from the cochlea stable. So this was used during development of the cochlear implant. And those of you who have seen a cochlear implant will recognise that this is almost entirely borrowed from cochlear technology, including all the attachment methods, the cables, which had been previously developed. So that was a very smart move. What it enabled them to do was get very well-tested technology into a device which was extremely cochlear-like and get it into clinical trials without too much fuss and bother. So the percutaneous plug allows you to connect directly into these wires and control the electrodes remotely just by plugging it in. That gives you your widest experimental space anyway. You can pretty much conduct any experiment you can imagine with that device. The surgical approach, reasonably simple, although there was a lot of development that went into this, as you can well imagine. Penny Allen, a vitreoretinal surgeon at the Eye and Ear Hospital, oversaw that in collaboration with Jonathan Yeo, another consultant there. A small incision is made behind the ear. There's a trocar that tunnels this device under the skin through to the eye. That little elbow bend that I showed you there is notched into this little notch in the orbital bone and secured in place. A scleral incision. The device is slid into that incision, sutured onto the sclera, and the wound is sutured closed. A long process, but it turned out a very effective one and a very safe surgery. One of the probably brightest outcomes and one of the brightest hopes for this device is that compared to other devices around the world, this has a very safe surgical approach. There they are, Murray, Morris and Diane. So these were the first 3 people to receive the Australian bionic eye. So Diane was first, a remarkable woman, and you couldn't ask for a better first patient. She got her PhD during her experience in social sciences, wearing this bionic retina, and has written a book, which I'll flash up in a couple of slides. And then there was Morris, and then Murray. We'll hear from Diane about her experiences.
It's been 15 months since I've been implanted, not really knowing what the journey was going to be like, but excited to be a part of it. What we've done in the testing has been amazing.
It felt wonderful that I saw this flicker of light that looked like a candle light that I probably blew out when I was 18, and I've never seen one since until 12 months ago, which was just wonderful.
Yes! You. Really? Yeah, just like a little quick flash.
We've now started working with the camera and working out how to move my head with the electrode to look at the outlines of the object, where it's an object and where it's a space.
So you can imagine, I think, and certainly in the research that I've been involved in terms of Eureka moments, this was just loaded with opportunities for Eureka moments. And to have moments like that where you're all sitting up in a gallery and there's the 2 people down in the audio-visual lab turning these people's devices on for the first time, and they start at low currents and then slowly go up on one electrode, and then you get that yes moment. And it's always pretty unbelievable, and you can hear the people in the room going, really? Because the patients also want this experience, you know, they really want it to work, and so you need to be very sure that it is in fact working.
So today Diane's doing experiments where we're having a set of obstacles in front of her on the ground, and she needs to move from the back part of this environment through to the front part, and we're looking at how long it takes her to pass through that pass and how many collisions she has with obstacles or brushes against the wall.
Oh, this is, you can see a reason for the bionic eye. You can see that there's a practical use for it. It's just, it's really good. Well, it's a different sight than what I had before I lost my vision. So it's learning to trust what I'm seeing, and that's why I say the more I'm doing it, the more I'm trusting what I'm seeing, and learning to see what I'm seeing is true. That's what's there. The more I'm doing this, the more I feel like I'm looking out of my eye. So I know there's a camera there, and I know I'm scanning with the camera, but it's like the image is coming through my eye.
So a couple of really important things in that little snippet there. Firstly, that movie illustrates how we did our data acquisition. So you have when you have a retinal implant, you have a couple of options. Either the implant itself can detect light coming in, so it might have photodiodes on it, or even little photovoltaic devices, as some of them do. So they operate more like a real retina. They interrupt the light coming in, they respond to that light, and they stimulate accordingly. Or else you can have a camera system where you have an external camera that takes information through some sort of visual processing, and then that goes back into the implant and you can stimulate accordingly. The photovoltaic approach has advantages, that the device is obviously smaller and sits purely in the intraorbital space, and it moves naturally with the eye. But the advantage of having a camera is you can pull all sorts of visual tricks with the information that you take from your camera. So it can go through into post-processing and do all sorts of interesting things. So did anyone recognise the particular camera that Diane was wearing in that last slide? You don't play enough video games. So that is a Kinect camera. It's a depth sensing camera. So rather than using intensity-based vision like you and I are using now, bright things appear bright, she was experiencing depth-based vision. So close things appear bright, which you can imagine is a useful way to perceive the world if your vision is very limited, which in her case, where in this device it still is. This is the book that Diane wrote about her experience. So yeah, a marvellous read if you're interested in what it might be like to go through a scientific study, especially one that attracted a lot of media attention. Where to next? Right, so one of these stimulation spots we refer to as a phosphene, a bright spot that's given to the patient through electrical stimulation. Diane had 24 electrodes, so this is a Melbourne scene depicted in 64 electrodes. Anyone want to make a guess? 1,000 electrodes? Someone got it? Yeah, exactly. So just very crudely, that gives you an idea of the kind of resolution you need to get to in order to get some sort of idea of what you're actually looking at. So that what Diane can do with her device is she can scan left and right, and she can sort of find the edges of things. So she'll get this perception that something is there and not there. And so they're constantly having to move their head like this in order to find the edges of things, which the people with RP that are profoundly blind report that they find very rewarding. Just the experience of having vision again is great. One of the things that Diane mentions is that she's having a conversation with someone that she can tell if they've left or not, which is apparently something that happens quite a lot. So she can still tell that the person is in fact in front of her still. But if you want to recognise who that person is, or you want to see whether they're smiling or responding to you, you obviously need a lot more electrodes than 24. So that's the BVA. Right from the outset, we're developing two devices. This one was designed to go into clinical trials early and prove that the suprachoroidal space could work. And the second device, which was the one that I was involved in, is designed to give higher resolution vision. So we decided to go for this epiretinal location purely because that is the closest point to the ganglion cells. So you can imagine current spreads quite readily in the body. If you want to stimulate nerves with high resolution, you need to be close to those nerves. This is the device that we built. So it is almost entirely fabricated out of diamond. So this is a diamond. This is the electrode array part of it. What you're looking at is the inside of it at the moment. There's all sorts of gold wiring which is inlaid into the diamond there, a few capacitors for power. There is a diamond electrode array on the front face. There's an ASIC which was designed by Nectar at the University of Melbourne, which is a standalone stimulator. So every channel on that ASIC is an individual stimulator channel capable of stimulating up to 3.3 volts, fully programmable so you can get a really wide range of experiments that you can do with this chip. And I think that was really instigated by the Cochlear people that were involved in this project, because one of the major lessons that was learned through Cochlear is you build the device as well as you can to be as safe as it can be, and the big advantages in the performance of that device came in post-processing and stimulation strategies. So what you do with those electrodes and how you process the sound information going into the device. So they built the ASIC with as much program flexibility as they could in order to make a device that you can just do a huge number of experiments with. And what that kind of meant was that we sort of had to work backwards. So the pitch of those electrodes is 150 microns. Each micron is about 120 square. And that was dictated because the chip already existed when we came in. So I think in some ways it might have been nice to work from biology back, kind of maybe look at the way that the retina and the neurons in the retina work, and take your spatial resolution from what you think you might be able to achieve from that end. The way we went is we went from the ASIC first, and the pitch of the electrodes was pretty much based on the lithography that they used and just the footprint of the stimulators that they used. So they told us, basically right from the outset, we want 150 micron pitch. So that's what we built. There is a diamond box that goes on the back, and then in order to make this thing soft enough to go into the eye, the whole thing sits in this silicone housing. We took basically the suprachoroidal component and put a magnet in it. And so that part slips into the suprachoroidal space. The epiretinal component comes in, and the two magnets locate the device in the area centralis, so the high-resolution part of the retina. Right, a little bit about diamond. We grow our own diamond at the University of Melbourne and we buy it. In terms of making diamond electrically conducting, because we wanted both here, we've got insulating diamond for the can, but we also needed conducting diamond for the electricity to get through to these stimulating electrodes. And we also needed the electrodes to be made out of diamond, so we needed conducting diamond there. Making diamond conductive you can do in one of two ways. So here's the face-centered cubic stacking of carbon atoms in a diamond. If you want to make it conducting, you can put an odd atom in, like boron, that changes the electronic structure of it enough that it becomes electrically conducting. We didn't use that. We used a type of diamond called nitrogen-included diamond. So all of these diamonds are polycrystalline diamonds, not single crystals. So there's millions of micro-sized or nano-sized crystals all packed into a film. Turns out when you grow them with nitrogen in the mixture, you can just essentially widen the boundaries between all these grains and make them very graphitic. And so in our particular type of diamond, the electricity is not conducted by the grains themselves, it's conducted in these sort of inter-grain spaces. This is what the actual pieces of the device look like on a €1 coin. So this is 4 millimeters square. The gold panels on the back there, there's the ASIC, beautiful piece of tech that. Amazing. SEM Images there of the square electrodes that we went with in the end. This sort of round circular section on the top was the best effort we could make at rounding this to the inside of the eye, or at least gave it a tiny bit of curvature. We came up with ideas later on about how we might have made that more round to fit better, but by then we were under a design lock, so we couldn't go with those. Some closer pictures there. This is some nice technology that we developed. This is using gold-based alloy, so we can mill, all of the milling of the diamond is just done by laser milling, and we can melt these gold materials into the diamond and polish them back, so you actually get all of the wiring and contacts is buried in the diamond, makes it very strong. Quite, because they're deep, they're high conductivity as well, low resistance. This is our NuNCD, or nanocrystalline diamond material on the top, and the ASIC. So one thing that's important here is that when you get up to really high electrode counts, you've always got this problem of how you get the information into your implant. If you were to take the approach they've taken with the wide view device of just having a wire that goes into each electrode, straight away you've got 256 wires going through the wall of the eye into the electrode, so it rapidly becomes untenable. So that cable is just too big. If that's going through the wall of the eye, it's definitely going to leak. When you have your stimulator directly attached to your array, you can do your stimulation. All you need is a few control wires and a power wire going to your device, so you can keep your cable very, very small. So this was an important part of the idea, and probably the main reason why we ended up using diamond. Because if you are putting your ASIC on your electrode array, the electrode array has to be hermetic, it can't leak. So everything here has to be encapsulated hermetically so that it's protected from the body and the body is protected from it, as I'm sure you understand. So we needed a way to make this electrode array functional and hermetic in order to accommodate this. When you're getting to high electrode counts, you just can't have any leakage at all. So these have to be extremely, extremely reliable feed-throughs. In basically 1 or 2 steps, we achieved this quite early on in the program. Late 2010, we got this working. If you start with a polycrystalline diamond film, drill a whole lot of holes in it with a laser. So this is the insulating diamond we've got here, the outside of the package. And then we grow our conducting diamond along the top. It's grown in a very high energy plasma, which means the diamond grows all into the nooks and crannies. There's basically no seam between the 2 types of diamond. Finally, we take a last pass with the laser in order to isolate all of these electrodes into the checkerboard that you saw. So this way, you've actually got a diamond in diamond conducting feed through. You can grow this for a certain amount of time, say overnight, and if you take it out in the morning and test it and it's still leaking, you can still pass helium through it. You simply put it back into the reactor, give it another day, and eventually all the holes will close up and you'll have yourself a hermetic array. So a nice, reliable, and extremely testable system. This is an SEM image that's been cut through with a focused ion beam. So we are just looking here. The red lines indicate where the conducting diamond is, as opposed to the insulating diamond, which is over here. We've never seen a gap in this diamond, which is why I think they're just so reliable. They're really very, very, very hermetic. They're quite high impedance, which means that they are often not suitable for recording where you need a really low resistance path through to your equipment. But because we're stimulating, you can basically push past that resistance just by upping the voltage of your stimulator. And we also, for each actual electrode, we had not 1 feed through, but 9. So you just put them in parallel. That divides your impedance of your feed through by 9. It's a really interesting material. So when we grow this, just out of spec, straight out of the reactor, it has this extraordinary micro and nanoscale roughness. Turns out this is one of the most biocompatible materials, well, I should say non-cytotoxic materials around chemically. It has, nerves seem to have an affinity for it. Some groups report that you can grow neurons on this without having to use laminin or fibronectin, for instance. So it's kind to nerves. It is, of course, extremely hard and quite abrasive. So in environments where you're going to get a lot of movement of your device, this is clearly going to be problematic. When you're stimulating, it's very easy to do electrochemical damage to your nerves. So one thing that you need your material to have is a high electrochemical capacitance. So what that means essentially is that you can put a lot of charge into that electrode only using a very low number of volts. And using high voltage in vivo is where you get the problems. This is where you start to do electrochemical damage to the body. So it turns out that this material does have that. In 2011, we had a nice paper going out where we showed we could grow a particular type of this diamond that did have sufficient capacitance to operate safely. And those safe limits are well established from other devices that are around the world. So this is kind of what it looks like, this stuff. You can imagine these grains of diamond are 3 to 5 nanometers in size, so extremely small, held together in this graphitic amorphous matrix. We do some chemistry to modify the tops of those with oxygen functionalities, and we do a little bit of etching so that the graphitic material is etched back away from the grains, and that increases the surface area. So we make a really rough material with high capacitance. Potentially nerves are going to like it, and it has the capacitance to stimulate nerves safely. You can ignore that. I just put it up because it was done on the synchrotron, so it was an extremely difficult experiment to do, so please appreciate the time it takes to get all the way out to... Yeah, right. Brazed metals. So, diamond has wonderful properties. It's extremely hard, it's extremely hermetic, it's very, very biocompatible, it's obviously going to last forever. However, you cannot melt it, you cannot weld it. So, in order to make this whole idea work, we needed a way to stick diamond to diamond. It turned out that there is a whole line of chemistry of metals where they're interested in sticking diamond grit to tungsten carbide pieces of metal in order to make cutting tools, like the grinders that you put in your angle grinder for cutting through bolts at home and all this sort of stuff. So luckily, people had already done this work for us. Unfortunately, most of them are based around silver. ABA means active brazed alloy. So these have got some chemical component that has a reason to bond chemically to diamond. It's usually titanium, forms a titanium carbide bond, so these metals will stick. So this stuff is fantastic, wets diamond beautifully, sticks like crazy, extremely strong bond, but of course silver is about as cytotoxic as you can get. So in the hope for some miracle, we just thought we tried it right at the outset and thought if it is an alloy, maybe it'll have different properties, but it didn't. It was very, very toxic. So we had to go with a gold-based brazed alloy. This is the makeup of this particular alloy, almost all gold, tiny bit of nickel, and a very, very small amount of titanium. So this, you can make it work through a lot of trickery, but it's just nowhere near as friendly to use. However, it turned out to be quite biocompatible, so we did back muscle implants in guinea pigs with this for 6 months and got a similar sort of immunohistological response to medical grade silicone. So that was our go-to guy, really, gold ABA. We use it like this, in one of two ways. So our polycrystalline diamond substrate, insulating diamond, laser milled trenches into it, simply melt the brazed metal onto the top, polish it back. If you stop at this point, you've got a wire. If we then cut through the whole assembly, you've got a gold edge. I mentioned before that the diamond feed-throughs are quite high resistance. So for our data and power lines that are going in supplying the chip, we needed a low resistance feed-through. The gold braze turns out it sticks very well to platinum as well. So we could simply drill a hole all the way through the diamond, feed through a platinum wire, melt the gold braze down inside it, and seal the platinum wire hermetically into the diamond in this fashion, so that you get a sub-ohm feed-through in the diamond. This was a lot less reliable than the diamond, but then we only needed 6 feed-throughs, so the chances are we could always make a hermetic array if we spent enough time on it. So this gold edge, feed-throughs for data and power, yep, got them. We've got this gold edge here. So that allowed us to put gold edges on both of our diamond slabs, put them face to face, and simply run around with a laser welder. So here's our hermetic seal for joining the 2 bits of diamond together. This was good stuff, really. This was Sam Lichter was one of my PhD students who undertook most of this work, including commissioning the laser gear, an amazing student. And this worked. It was difficult. Our yield is still low. We're still optimizing it. And I think mainly that's just because of the scale that we're working on. I mean, this is a 40 micron wide weld seal, so it's extremely, we're working right at the tolerances of the equipment and the particular weld. So I think if we go a bit bigger, probably a lot of those problems will go away. And we're now working with the smart stent people who are doing intracortical stimulation on a can, which is a bit bigger, and I hope that's more successful. Some nice SEM images there of the feed-throughs. So these do have a platinum wire in the middle. For some reason, platinum and gold don't really differentiate on the SEM. And we now have this kind of winged design here just to lock the feed-throughs in rotationally a little better. Oh, just some histology there. You probably understand more this more than I do. This is a H&E stain. The blue indicates scar tissue around the device. So here is a negative control. This was chemically modified to be toxic. So there's your negative response. Polycrystalline diamond and silicone, got a sort of a similar response. And then this very fine line here is the NUNCD. So Richard Williams, the pathologist over at St Vincent's, is very impressed with this material. He says it's about as good as he's ever seen for an implant, which is great. Doesn't degrade in any way, so it doesn't seem to dissolve or disappear. So we're quite positive that gold ABA is going to be really the material we want to use for these devices going forward. Right, just step aside for a second. before I started my PhD, I was down to the last 2 candidates to work as an electrochemist at the TY Point aluminium smelter. These are the anodes I would have been working with there, carbon anodes. Huge things. So, luckily or unluckily, depending on your perspective, I did my PhD and ended up working with these carbon electrodes, 200 microns in diameter. So this seminar series is about translation of research. So essentially, most of the experiments that we have to do for a biomedical implant are about proving safety, number one, and efficacy. So this experiment was really an efficacy experiment with a little bit of safety tacked on. We needed to show that the particular type of diamond that we can grow can stimulate, A. We always knew we're going to be able to stimulate. If something's conducting and you put it next to a nerve and you put electricity through it, it's going to stimulate. What's in doubt is whether you can stimulate without killing that nerve. So that's the other part of this experiment. So this is a lovely experiment, actually. We conducted it over at the National Vision Research Institute. They have a technique called whole mount, where you take a retina out of an animal, usually a rat or a rabbit. They keep that retina alive in a dish for up to 4 to 5 hours, and they can stimulate on top, and they can patch clamp record from ganglion cells in that retina. So in a real piece of tissue that's operating normally, relatively normally, you can actually do this type of experiment. They can then backstain that particular ganglion cell and tell you exactly what type of retinal ganglion cell you were working with. One of, I think, 24 identified retinal ganglion cells in rat retina currently. So we were able to put this down onto the retina, get a patch clamp in, find a ganglion cell adjacent to our stimulating electrode, and you can stimulate, slowly increasing in current, and then find out when this adjacent retinal ganglion cell starts to be activated by that stimulation. So it's a sort of a first pass. It's obviously not telling you whether that signal would have gotten all the way back through the optic nerve and been a phosphene, but it is telling you at least that you've activated retinal ganglion cells in a real operating retina. And that worked well, and it worked within voltages that we were happy with, around about 0.5 volt. So you tend to not get too much bad electrochemistry happening in the body at 0.5 volt. That went to biomaterials, nice paper, good result, and really good collaboration with the NVRI. Then we go to in vivo experiments. So these experiments are constructed like this. We have a standard off-the-shelf cortical recording array, which goes into the visual cortex. Our device, this experiment was conducted in cat. We have our device implanted against the retina of the animal. We stimulate here, same sort of experiment, increasing current, and then recording from the visual cortex in the animal, we're looking for a corresponding activation in the visual cortex responding to the stimulation. Here we were really interested in the size of our electrodes and whether we could get a response off just one electrode, because they're about as small as anybody in the world has tried, and they're certainly closer together than anybody else in the world has tried. So the experimental result we were really chasing was stimulating 2 adjacent electrodes, one after the other, and seeing some sort of shifting image in the brain. So we were finding some proof or evidence that we were providing higher resolution than you would normally get. As a spoiler, we didn't get that result, but we did get some results. Surgically similar to the suprachoroidal implant, we put our back magnet in. This is a complicated surgery, putting the epiretinal implant in. The surgeons have to come in with a little cutter here with a vacuum on it. They need to cut away the entire lens. This is done with a port in here which keeps the intraocular pressure constant during the experiment so that there's no damage done through high or low pressure inside the eye. Once they've taken away the lens, just to get it out of the way basically, they make a full thickness scleral incision after a bit of diathermy to cauterize it. Here's your incision. The device gets put through here. There's a patch on the outside that gets sewn to the outside of the sclera, and that sort of seals up the wound. And voila, you've got your device located inside the eye against the retina. This is what you see when things go well. So this is our stimulation time. And then after some latency, usually around about 6 milliseconds, this is typical, you get a train of spikes. And depending on the amount of charge that you use, you'll get more or less spikes up to some saturation amount. This is a full current, which in this case was around about 20 microamps or above. You get this number of spikes, and that's where it kind of stops. So you get this kind of grayscale curve. It tells you how much current you need to put in that electrode in order to get the maximum response of this electrode. And the smaller current is better, obviously. You want to be able to use the lowest current you possibly can so that this device is as safe as possible. This is the star result, of course. I'm not going to show you the worst result. The worst result is noise up here, and we've got plenty of that as well, as you can imagine. Very, very, very difficult experiments to do, just to find the location in the cortex where you're actually getting activation, because this device is only actually a very small window in the visual field. So finding that area in the visual cortex is fairly large is extremely difficult. So the majority of our experiments, we got nothing at all. And that could have been because the device wasn't working, or maybe the implantation didn't go well, but most likely it's just that you never found the area of the cortex where you were getting activation. It might have been too deep, you couldn't get your electrodes to it. So really difficult experiment to do. These are the results that the three results that we did get trying this experiment. This is essentially what we got. Experiment 1, Experiment 2, Experiment 3. So these are the threshold charge. This is where we got 50% up that curve, so halfway through the maximum saturation. This is the spread of results for all of the electrodes that we tried on that array. So this is our safe limit here. So our first set of data showed that all of the thresholds were above our self-imposed safety limit. The second experiment that was successful, we were getting closer, and then our best experiment, we had all bar 2 electrodes down in the safe zone. So I guess we're coming to the end of the funding for this project now, right now. This is clearly not enough results to say that this array is effective and safe. However, we feel it's enough to say that there is some hope for this, and we should carry on. We certainly haven't proved yet that you even can achieve this sort of resolution in the retina by stimulation. We have no evidence that stimulating one electrode here and 150 microns separated is giving the cat any different experience in the brain. And the arrays around the world that people are wearing that have similar resolutions, they tend to be not getting any particular benefit over and above the much lower resolution arrays. So there's still actually doubt whether the strategy is even valid. Can you in fact get better resolution by going smaller? Yeah, a lot needs to be done. I think most of the stimulation strategists are playing around with the idea of opposing stimulation and trying to get the current really focused in on particular sets of nerves. And that may be where we have an advantage. We have a chip that can kind of do that stuff. Yep. The magnetic attachment. I think this is a great idea. I really think it's got some promise, but however, in the particular space we were in, really difficult. So this is a nice X-ray here showing the magnet on the back of the device, all of the internal wiring of the device, and the magnet in the suprachoroidal space. Diamond is invisible to X-ray, so all you get is the metal, a view from the top there. So it does work, it gets in, it gets located. You can see there's a bit of an offset though. So the design we chose, the magnets didn't lock into just one location. There's a couple of sort of nodes, so that needs addressing. The magnet tended not to sit completely flush. So up here, we're looking, this is an OCT image. Here is our magnet. We can't see through the magnet. We can't see through the diamond with OCT, so we could only look at the silicone wing on the side. This silicone wing up here is sitting fairly flush on the retina. It looks good. However, if we look at the other side of the device, you can see that this silicone is impinging quite markedly into the retina. So this device just tilted in one corner, and that could have come from slightly off magnetic alignment. We've only got 40 microns to play with here in terms of tilt, so the tolerances are really, really low, extremely difficult. Just fundus images straight through the pupil here, looking at the device, 2 weeks, 6 weeks, and 12 weeks. And despite being magnetically located, it's clearly moved. And we've got this bruising and this discoloration in the retina here. So we we like the idea. We haven't got it right yet by any stretch. And I wonder whether this is just too delicate a space to be that rough. I don't know. Well, it would have to be very, very perfect in order to work sufficiently well. And there's the, once this eye was dissected, here was the results we got here. You can see healthy retina here. We still don't have the histology for this. It's a very new result. But the retina is being completely decimated down in this corner where you saw that silicone tipping in. So this is the kind of safety stuff that goes on with biomedical implants. It's very much have a good idea, try it within the constraints of your budget and your time, and then a lot of post-analysis to show that you're not doing any damage. Safety first, efficacy second. Where to next? Right, so funding has officially run out. The Bionic Vision Australia and Bionic Vision Technology, which is the company that have been born out of that, have decided to go with the wide view device exclusively going forward. So they're only seeking further funding from venture capitalists for that. All of us are writing grants through the regular granting bodies, and Stephen Prower, who's my boss, who was scheduled to talk to you today, but he's in Canada at the moment on a sabbatical. And he has apparently started a company over there, and he's got some investors that are interested in our technology and some spin-offs as well. The diamond hermetic encapsulation is something that's got possibly quite a lot of potential, and they're interested in further developing the retinal stimulation array that we've developed. That's us in a nutshell, 4 years of effort. I think a very certainly from the wide view, a remarkable feat to get from concept and grant in 2009 through to the end of a first clinical trial after only 4 years and heading into the next one is astonishing. I'm not aware of anything else, and you may be, that's gone that fast. For us, I think we fulfilled our mandate in terms of building a diamond capsule, getting a chip into it, building a stimulation array out of diamond. We've done that. It is hermetic. We don't know if it works for retinal stimulation yet. It may do. And we haven't solved the problem of attachment, so there's plenty of work left there to do for us. Lots of people involved in this project, as you can well imagine. So my primary desk sits at the University of Melbourne. I also have a desk over at the Bionics Institute, where I'm an honorary fellow. So all of the in vivo work gets done at the Bionics Institute. Very close work with the National Vision Research Institute. So they do all that whole mount retinal stuff and are really interested in the science of vision and how ganglion cells work. ARC, this whole thing was funded by a special research initiative, and I work closely with the materials people at the University of Wollongong through Gordon Wallace's group in terms of understanding and characterising materials. Thank you for your time.
Fantastic talk, and certainly, from our perspective, the insights both into the trials as well as the tribulations of this technology, the amount of work that needs to be done for the outcome is quite remarkable.
Yeah, well, I mean, we were so gung-ho at the beginning, as you know, a lot of us were new postdocs, and you're coming to the end of 4 years. It's an enormous task. Just proving the safety of these things, as you can well imagine, and a lot of those experiments are just very, very time-consuming. Lots of surgical hours. Lots of animals and, yeah, big projects.
Can you see an application for this technology beyond retinal stimulation?
Yes, aspects of it. So the diamond electrode array, and in fact that's the crux of my research going forward, is branched out from a lot of this technology. So I'm looking at replacing the diamond electrodes with graphene and carbon nanotube yarn electrodes, so something that's a lot softer. And we're interested in going cortical. So these are for devices. It turns out that these particular fibres that I get from Wollongong are reasonably good at both stimulating and recording. And there's a big drive for these sort of devices generally to be what they call closed loop. So these are devices that can stimulate, and then they can have some sort of feedback function to tell you whether that stimulation was successful, or else they are brain-machine interfaces that say control a prosthetic limb, but there's sensors on there that give you feedback in terms of touch. So this is a closed-loop system. So But that will use essentially the diamond base, the feed-through technology, and the ability to put the ASIC inside the can. So you have a complete remote system with its own control that can be wirelessly accessed just as one little discrete unit. So I think there's lots of it that will spin out into other applications.
I'll ask David this question. ASIC.
Application-specific IC, so a chip.
Chip. Questions from the audience. Thank you.
And 2 questions. Really nice presentation.
Thank you.
First question is, when you're placing it intraoperatively, and would ophthalmologists use any stereotaxy or any navigation to ensure you're putting it right on the fovea, or do you just put it there and wherever it sits, it sits?
Throughout this whole process, there was lots of talk about that, getting much more control over where it goes. We never got there, so it was always eyeballed, basically. With the magnet, you put that suprachoroidal array in with as much control as you can get, but often during the healing process, because we would do those surgeries about 2 weeks apart, so the suprachoroidal would go in, you'd allow that to heal, and then you'd put the epiretinal in. So you put it in, and you do your best guess. You can kind of see the magnet through the choroid, but it would move during healing. So yeah, really, really very tough. I think in the end, one of our plans was to maybe go with a sandwich system, so the suprachoroidal array went in and you could lock it somehow, and that might be scleral sutures that go through dacron patches or something like that. And that if you can lock that, and then if the epiretinal component is somehow locked to the suprachoroidal component, then you can start to get closer to exactly where you need to be.
So you need to calibrate it for each patient depending on the position?
Potentially, yeah. Yeah, because eyes change quite a lot. And especially with RP patients, because the longer they've had the disease, they get quite a lot of degeneration of their retinas, so their retina structure can change, and yeah, things do move around. And their ganglion cell matrix kind of, the act of seeing organises your retina as you grow, and when you lose it, your retina deorganises over some time.
The other comment I had actually was that, and in vivo, and your retinal cells are obviously all firing to an extent, they're just not firing significantly enough. So your arrays, would they be all quiescent until there's a stimulus? Because I was thinking that what if you could increase the resolution? There's I can't remember the name of the theory, and it's been a while since I've been neuroscience, but if you had a single point of stimulation, you've got a baseline activation across your entire array.
Yeah.
When you have that point of stimulation, you damp down stimulation in a ring around your primary electrode, and maybe that will increase the I guess the perceived and specificity of that.
Yes. So 2 parts there. The first part is the issue of fade, which is when you overstimulate a ganglion cell, it needs a recovery time. So if you do overstimulate, you go at too high a rate, that the brain will simply say, ooh, that's not right, and just turn it off, and the cell can't recover, and you end up... It's the same basic phenomenon, slightly different. Actually, I'll cut that comment. But fade, you can, but there is a rate, and that's quite well established, and if you stimulate slowly enough, the nerves have time to recover, so you can get around it that way. The second part, I think you're talking about putting a guard of electrodes around the stimulating electrode that kind of do something electrically to promote that one. And yes, and in fact, UNSW, who were very involved in this project, have got patents surrounding exactly that in terms of stimulation strategy. So you can have, and you can have both of the electrodes stimulating, but the amplitudes of the stimulation is opposite. So you can potentially suck charge density out of one area in the guard and push charge density into the middle bit, so you can get more specificity in there, less current spread. Yeah, and you can essentially do a Fourier transform across these things, so you can have all of the electrodes stimulating simultaneously, so they build up a wave, and you can actually create patterns of electrical field on your electrode. And these are all the things that we hoped this device would be capable of doing once it gets into clinical trials, if it gets into clinical trials.
Yeah, really exciting. What the people who are in the first clinical trial, is their device put in such a way that it can be upgradable as new versions come online?
No, they were required to have their devices removed after their, which is kind of a little sad for them. So I don't think they're precluded from having a different device in the other eye, but I think part of the safety consideration for them was that they would be tried for 12 months, which was then extended to 18, and then the device had to be completely removed. So they're all explanted now. Part of the reason for that is they needed to look at the devices and see how they fared, how the electrodes look, have they degraded, have they dissolved, that sort of stuff. Have any wires broken?
One last question.
I just wondered if maybe something like a subretinal implant would give you advantages that you don't get with your epiretinal implant. So you'd be less likely to damage the retina, I would think, so less likely to crush it. I would have thought you would get a lot more stable implant.
Yeah, and so there are one major group in the world, EpiRetina, have a subretinal device, and that seems to be performing relatively well. And surprisingly, theirs is actually one of the photovoltaic arrays, so the light goes all the way through the retina and then hits gates on that array and stimulates accordingly. Surgically, it's very difficult to get to that space, just because the retina is so friable, and the choroid actually supplies most of the blood supply for the retina as well, which means that you're restricted to a very small device, so probably around about the size of ours, and it also has to be exceedingly thin, which means that you don't have a lot of room to put complicated electronics in there. So it's a better location in terms of implant stability, surgically troublesome, and it places limits on the technology you can put in that space. But yeah, pros and cons. But their device, the EpiRet device, is working pretty well. They've got the record at the moment for electrodes, 1,500 electrodes on their device. But because they're all PV controlled and they don't have any programmability, they tend to get big patches of electrodes stimulating simultaneously. So in fact, they're getting no better performance than the devices that have 20. And that's that lack of programmability that's at fault there, really.
Okay. Thank you very much.